Professor Holland kindly did the transformation experiment for us on the weekend. So we've got this incredible transformation experiment described in chapter 15 that we're going to share with you. Professor Holland board and okay, they're hiding. You will find the transformation plate. It's probably right in front of you, right? Transformation plate. No, looking right at them. We'll find them, guys.
Oh,
We'll find them. We'll find them. Okay. So let me first describe to you the procedure for transformation, which is described in chapter 15 of your lab manual. And you guys do remember that Tuesday, which is our last day of lab, so not this Thursday, but next Tuesday is our last day of lab, we have that control of growth quiz, and it will include this discussion of transformation. Okay, so you make sure that you understand what's going on with transformation. Okay, and again, it is described in chapter 15 in your lab manual. That'll be your reference. It's also great because it's one of the examples of horizontal gene transfer, which will be on Thursday's exam. So it's good. Some of our discussion here might help you with the questions on our Thursday exam. Okay, so transformation, you guys will recall, is one of the three ways that bacteria can transfer genes horizontally. The others are conjugation and transduction, right? Okay, so in transformation, by definition, transformation is the uptake of naked DNA from the environment by a competent bacterium. And you guys recall from lecture, we said that some bacteria in nature are naturally competent because they have the genes for DNA binding proteins. So in nature, some bacteria, like Streptococcus pneumonia, will synthesize these DNA binding proteins. It permits them to bind to naked DNA in the environment, and then they can pass that DNA into the cell. Now, with E. coli, and you guys know that E. coli is kind of our workhorse of microbial genetics, E. coli lacks the genes for DNA binding proteins. So that means that E. coli does not become competent in nature. But through some incredible research and experiments, it blows my mind that people figured these things out. Scientists were able to devise a way to artificially make E. coli competent. So, one of the questions I will surely ask you on the quiz is, how were the E. coli treated to make them competent? And the answer is, we treated the E. coli with cold calcium chloride. Okay, so again, you guys, I mean, not to make you silly, but I know we're all tired. So, what was the treatment we used to make the E. coli competent?
Cold calcium chloride.
Cold calcium chloride. Excellent. Now, the actual changes in the E. coli cell that makes them competent, I think we're not exactly sure what's going on. So I just drew this diagram showing a cell wall cytoplasmic membrane that has some pores in it. And one thought is that the cold temperature helps stabilizes maybe transient pores in the outer membrane, transient pores in the cytoplasmic membrane. Sounds a little wishy-washy to me, but anyway. And then the one purpose of the calcium chloride is that when it ionizes, it releases calcium cations that will bind to the negatively charged DNA that we want to pass through the cell wall, past the cytoplasmic membrane into the cytoplasm of our bacterium. So by having calcium ions bind to the DNA, it will create an electrically neutral molecule that will have a much easier time passing into the bacterial cell. So that's one model of why the cold calcium chloride might work. Okay, so Professor Holland then treated our strain of E. coli, and it's very important that the initial strain is ampicillin sensitive for this experiment, and we'll explain why in just a bit. She treated her E. coli with cold calcium chloride. That made them competent. And then she mixed the competent E. coli with naked plasmid DNA. Naked meaning it wasn't enclosed in another cell, it wasn't enclosed in a bacteriophage, right? So the naked DNA that she mixed with her E. coli was plasmid DNA that was commercially prepared. And from the manufacturer, we know the genes of interest on the plasmid DNA. So this is you know, we're changing our size here, you guys. This is a cartoon of our covalently closed, this is our cartoon of our circular plasmid DNA. The plasmid is called P, the little p stands for plasmid, and then it's called pGreen. So whoever develops or isolates a plasmid, they get to name it. The reason it's called pGreen is that some of the genes on the plasmid encode information for a green pigment. So the E. coli that take up this plasmid when they grow and form colonies on plates, we'll see they actually have this kind of fluorescent yellowish green pigment to them. The second gene of interest on the pGreen plasmid is an ampicillin resistance gene. And it's important that you guys know that the ampicillin resistance gene, the actual product is the enzyme beta-lactamase. So what does the enzyme beta-lactamase do? It hydrolyzes beta-lactam antibiotics, such as ampicillin, right? Okay. So bacteria carrying the pGreen plasmid, they are going to make beta-lactamase. They will destroy any beta-lactam antibiotic in their environment, like penicillin or ampicillin, and thus they'll be able to grow in the presence of, for example, ampicillin. So we would call them ampicillin resistant. That will become important later when we describe how we're going to select for our pGreen transformants. So, as we said, the cold calcium chloride made the E. coli competent. Professor Holland mixed the competent E. coli with our prepared naked pGreen plasmid DNA. So the next question is, you guys, how did she get the pGreen plasmid into the E. coli? How did she actually transform the E. coli?
Heat shock.
Heat shock, right? And what heat shock is quickly increasing the temperature for a short period of time. And if we think about what an increase in temperature means, temperature is the average kinetic energy or average speed with which molecules or cells are moving. So by quickly increasing the temperature, we're going to increase the speed with which the E. coli are moving and the plasmid molecules are moving. And what we're hoping is that it will have a collision between one of the competent E. coli and one of the little pGreen plasmid DNA. It will have a collision that will permit the pGreen to pass into the interior of the E. coli. Okay? Now we don't want the heat shock to last too long because our E. coli are so delicate they could end up lysing. So usually this is a very short exposure to a 42-degree water bath for about maybe 60 seconds or so. Okay. So what we're hoping then is at least some of our E. coli got transformed. Most of them won't be transformed. This is a relatively rare event, but we're hoping at least a few of them got transformed with pGreen plasmid. What we then do is we add a little bit of broth. We let the cells recover at 37 degrees Celsius because this is traumatic. Can you imagine if you were put through this process? You'd be traumatized, right? So we want to give the cells a chance to recover. We also want the cells, we want the E. coli RNA polymerase to start transcribing the pGreen genes, and we want the E. coli ribosomes to attach to the messenger RNA and start making the gene products. And the most important one for us is going to be the beta-lactamase. Okay, so during recovery then, the cell wall, cytoplasmic membrane, they regain their integrity, and very importantly, because of the pGreen plasmid, what will this little E. coli pGreen transformant start making? Beta-lactamase, right? And they're going to make so much, they're actually going to start secreting it. Okay, so that's some of the events during recovery. Now, if you think about this, you guys, Professor Holland was performing this experiment in a little microcentrifuge tube. And let's say, we'll just make this up. Let's say there were like 10 to the 9 E. coli in that tube. And let's say only, oh my goodness, let's say maybe only, we'll make it up, say only like maybe 10. 10 of them were transformed. How the heck are we going to find those transformants? Most of the E. coli weren't transformed. If we just put them on regular media, we'd probably have just a lawn of bacteria, right? How could we select for the transformants, what kind of media could we use that perhaps would only permit the transformants to grow? Well, we'll think about it, guys. Okay, let's pretend we had some plates that had ampicillin in them. Okay, so let's say this is our plate, and let's say it's got ampicillin in it, right? And we'll use orange to represent ampicillin in the agar. Now my dimensions here on my cartoons are totally, out of whack. So let's say the orange equals ampicillin. Okay, and what I cartooned here is just a few of the E. coli from that initial transformation mix. And again, you guys, these aren't colonies. These are like huge, giant E. coli cells, so represent cell wall cytoplasmic membrane. This squiggle is a chromosome. And what we want to note here is that most of the E. coli were not transformed, right? It was a relatively rare event. So in this cartoon, 4 of the E. coli were not transformed. Only one of them down here, there's a little pGreen plasmid. We only have one transformant here. Okay. We're going to plate the bacteria on the surface of the plate and incubate it. What's going to happen to these guys that weren't transformed?
They'll die
They're going to die. Why are they going to die?
because they don't have the beta-lactamase in there.
Right, okay, so we want to remember, you guys, and again, good review for the final exam. Ampicillin as a beta-lactam is going to bind to the E. coli transpeptidase and block cross-linking of the peptide glycan of these little guys, right? So why are they going to die? They're going to undergo osmotic lysis, right? Because the ampicillin has blocked the ability of them to cross-link their peptide glycan, right? So these guys are going to growth. What about this guy? The guy that picked up the pGreen plasmid DNA, our pGreen transformant. Will this little guy die?
No.
Why not? What is it making that's going to permit it to grow on these ampicillin plates?
Beta-lactamase.
You got it, you guys. That's it. It's making all this beta-lactamase. Okay, so the arrows represent beta-lactamase. And what is the beta-lactamase doing to the ampicillin in the agar near that transformant?
Hydrolyzing it.
What's the beta-lactamase doing to the ampicillin? Destroying it? Hydrolyzing it? Yeah. So we'll show that in the agar where that transformant is growing, the beta-lactamase actually is secreted into the agar. We're going to create a little ampicillin-free zone, right? Meaning, ampicillin-free meaning there's no ampicillin present because the beta-lactamase from the transformant has destroyed it, right? Let's hydrolyze it. Okay, so now what we're going to do, you guys, is we're going to incubate these plates. We're going to incubate them. And now we're going to go from, looking at individual cells, we're going to look for colonies, all right? So we're going to incubate. So here's our plate after incubation. And remember, we've got, ampicillin here. Will we have any colonies where these non-transformants landed? No, because they died, right? So the only colony we're going to have is going to be a colony formed by the little clones of that initial transformant. It's going to grow and divide. It's going to grow and divide. And these plasmids, you guys, they replicate. They replicate. So we can have multiple copies. So whenever the bacterium divides, thanks, Francie. Whenever the bacterium divides, both the daughter cells will have a copy of the plasmid. So the plasmid is copied and passed on to all the daughter cells. Okay, so following incubation then, we will look for these beautiful green, yellowish colonies. They actually fluoresce under black light. They're really gorgeous. So now, you guys, again, we've changed our scale. This yellowish green colony is a transformant colony. It would represent maybe 10 to the 8 to 10 to the 9 of the descendants of that initial transformed E. coli. They would all be carrying the pGreen plasmid DNA, right? And because there's so many, they would be pumping out beta-lactamase, so you probably have a pretty decent ampicillin-free zone, right? So this is that ampicillin-free zone. And again, what is this transformant colony producing that's destroying the ampicillin? Beta-lactamase. You nailed it, you guys. So the red arrows again indicating the beta-lactamase. Now, what is so cool, and you guys, I apologize, let me just see if I can't find those plates, because there's the coolest thing going on with those transformation plates. So, if anybody finds a little tray, and it's probably right, the more obvious it is, the less likely I need to see it. Okay, let me be here. They've got to be here somewhere. They are just gorgeous. Transformation plate. No wonder I couldn't find them. Okay. What we'll do is we'll put these on a little tray. But these things are gorgeous, you guys. So what Professor Holland did is she ran all the controls. Okay, so she had one tube of E. coli that she treated with a cold calcium chloride. She heat shocked them, but she didn't add the pGreen plasmid. So that's the negative pGreen. And what she did is that mix, kind of her negative control, she plated that mix onto LB is a type of media that E. coli really likes to grow on. LB amp. Amp means there's ampicillin present. So she plated the negative control on the LB amp plates, and nobody grew, right? So that was an important control. Then she took LB agar that lacked ampicillin and plated the non-transformed E. coli to make sure that they were alive, and they grew like a Zolana bacteria, so that was an important control. This is the exciting one, you guys. Okay, so then she took her transformation mix and plated it onto LB agar that has ampicillin in it. And can you guys see that? You see those beautiful colonies? Those are the transformant colonies. And not only are they gorgeous, but something really cool is going on that really has application to what you guys are going to go into as far as medicine and dentistry. Around the little transformant colonies, you're going to see these little tiny white colonies. These little tiny white colonies growing. And these little tiny white colonies, they almost look like they're circling the transformant colony. And so they're called satellite colonies, like they're circling the transformant planet here. Okay. So this is such an important question, you guys. What or who is making up those satellite colonies? Who do you think they are? Who else could have been in this mixture that got plated onto those LB amp plates? Okay, so we have the transformants growing. Who else was in that mix? E. coli that did get transformed, right? So if these are the non-transformed E. coli, how is it that they're growing on the LB amp plates? We said they were going to be killed, right? How is this possible?
Transformation.
Okay, and if we presume they weren't transformed, right? So these are E. coli that were not transformed. How is it that they can grow on the LB ampicillin plate? Notice their location. Location, location, location. Where are they growing? Who are they growing next to? The transformant. What is the transformant making?
Beta-lactamase.
Beta-lactamase. The beta-lactamase is doing what to the ampicillin?
Hydrolyzing.
Destroying it, right? So does it make sense that these satellite colonies represent the non-transformed E. coli that were lucky enough to land close to a transformed E. coli? They were fortunate enough to land close enough to the transformant. The transformant started breaking down the ampicillin before all these little guys got killed. The non-transformed E. coli living next to the transformant colony, they're living in that ampicillin-free zone. Or it's probably agar that has just a reduced concentration of ampicillin, right? Now, why is this important, you guys? Imagine this is tissues of your patient. And let's say this is a what they call a polymicrobial infection. What do you think polymicrobial infection means? More than one type of organism is in there, right? And this happens often. It can happen with nosocomial infections. It can happen with traumatic injuries where, you know, dirt gets forced into a wound. You can have more than one microbe growing, right? Just imagine, you guys, this is your patient with a, you know, polymicrobial infection going on. The point here is that if there's a single bacterium in there that's pumping out an enzyme that can destroy an antibiotic, that single microbe can produce an antibiotic-free area, so the other microbes that lack antibiotic resistance genes, they might be able to grow there if you put that patient, say, on ampicillin. Does that make sense to you guys? Using your imagination, like if this is one of those polymicrobial infections in your patient, let's say you put your patient on ampicillin, if there's a single, we could pretend Staph aureus is in there pumping out beta-lactamase. That Staph aureus will be destroying all the ampicillin. So the other bacteria present that lack beta-lactamase genes, they're going to grow fine because the Staph aureus is destroying all the ampicillin for them. Does that make sense, you guys? Like how we can see application for this in the real world? Yeah, so remember, you guys, think of your agar plates. Think of that's your patient. Those are the tissues of your patient. This is the bacteria causing the infection in your patient. What kind of problems do we get into? If we have a single beta-lactamase producing microbe present, it can destroy the beta-lactam antibiotics you give your patient, so all those other bacteria could potentially grow. Yeah, so those are tricky ones to treat, those polymicrobial infections. Okay, guys, so the point here is, like, you know, I really would recommend that before you leave today, come up and look at these gorgeous transformation plates. Very important. Okay, here are quiz questions, you guys. How did Professor Holland make the E. coli competent? Cold calcium chloride, right. How did she actually transform the competent E. coli? Heat shock, okay. And just let me share with you that usually folks get the two mixed up. They switch them around. People will say, how were the E. coli transformed? And they'll say cold calcium chloride. That's wrong. Okay, I'll say, how were the E. coli made competent? People will say heat shock. That's wrong. So just be aware that that's confusing, right? Okay, why could the transformed E. coli grow on the ampicillin plates, but the non-transformed E. coli could not grow on the ampicillin plates? The transformants carrying the pGreen plasmid were making beta-lactamase. What does beta-lactamase do? It hydrolyzes the beta-lactam antibiotic ampicillin, right? And that's why the transformants can grow. Good. Okay. Why were the non-transformants killed? And what I'm asking you here for, just so you know, is I'm asking you what is the mechanism by which beta-lactams kill bacteria?
They like beta-lactamase?
They bind to bacterial transpeptidase and block the ability of the bacterium to do what? Cross-link the peptidoglycan, right? And if we don't cross-link peptidoglycan, what's going to happen to the bacterium? Osmotic lysis, yeah. Right? Okay. And the reason I'm doing this, you guys, this is stuff that's going to be on the final. So it's not only for the little quiz, this is stuff that you're going to see on the final too. Okay? Yeah. Sure. Okay, good. We'll do it one step at a time, okay? All right, so ampicillin, you guys. Ampicillin is a beta-lactam antibiotic. Beta-lactam antibiotics are irreversible competitive inhibitors of bacterial transpeptidase. Excellent. Okay. So if we expose our bacteria to penicillin or ampicillin, bacterial transpeptidase can't function. What won't occur? The peptidoglycan cross-linking won't occur. So what? If peptidoglycan isn't cross-linked, what's going to happen if the bacterium is growing in a hypoosmotic environment?
Osmotic lysis.
Osmotic lysis, right? They die, right? Okay, so that's the mechanism of the ampicillin. Step 2, and this is where folks get confused. Step 2 is to describe the mechanism by which beta-lactamases work. What do beta-lactamases do? They hydrolyze beta-lactam antibiotics like penicillin and ampicillin. They destroy it, right? So if we are bacteria and we want to survive on ampicillin agar plates, one way we could do it is to make beta-lactamase. Okay, so that would destroy the ampicillin and permit us to grow. Yeah, these are so good, you guys. And believe me, on the quiz and on the final exam, folks, it's confusing. The difference between a beta-lactamase and a beta-lactam antibiotic, that's really confusing. But you want to make sure you understand the difference. Okay? All right. Okay. So that is that. Now, what the second thing we're going to do, we have 3 things to do today, you guys. The second thing we're going to do is we're going to do control of growth 1 follow-up. And what we're going to do is have you pull your plates from the incubator. And first of all, you're going to take a look at your UV irradiation experiment. You guys will recall that you spread beautiful serratia marcescens over the surface of your plate. You took the lid off the plate because we know UV irradiation cannot penetrate the plastic. You covered half the plate with an index card to act as an internal negative control. And then you irradiated the other half of the plate for either 1 minute or 4 minutes. And what we were looking for was 2 things. We were looking for mutations caused by incorrect thymine dimer repair of our serratia marcescens. Remember, thymine dimer is the abnormal DNA structure that's formed following UV irradiation. So what you're going to look for on the irradiated side of your plate are smaller colonies and colonies that are no longer making pigment. Okay? And you'll also notice what? There are fewer colonies on the irradiated side, right? So that's telling us that UV irradiation also did what to our bacteria? You guys are so tired. You just look so pooped. Okay. Did the UV irradiation kill some of our bacteria?
Yes.
Yeah, we had huge kill. And if you take a look at your 4-minute irradiation plates, on many of the plates there's nobody growing on the 4-minute side, right? So that shows that UV irradiation can kill contaminating environmental microbes. So that's one reason why it's used in operating rooms when nobody's present. It's a reason that they're used in microbiology safety hoods to kill any contaminating microbes when people are not working. Okay, what is the risk if we expose ourselves to UV light?
Skin cancer.
Skin cancer and eye damage, right? So just remember that you want to protect your colleagues out there in the real world. Good. So you want to make note of that, record it in your lab notebook. And then the second thing we're going to do, and this is going to take some teamwork, you guys, but the sooner you can do this, then the sooner we can move on to the last part of today's experiment, and the sooner you can, go to lunch. And I know you're all looking like you could use some lunch or coffee or both, okay? So you'll recall the second part of the experiment was to compare different household antiseptics and disinfectants with their ability to inhibit or kill 2 different bacteria. One was a gram-negative bacteria, Enterobacter aerogenes. The second one was a gram-positive bacterium, Bacillus subtilis. Now, the way we're going to compare the effectiveness of our antiseptics and disinfectants is we're going to have you, I'm going to go ahead and borrow some of your plates. We're going to have you pull the disc diffusion plates, and we're going to have you measure the diameter of the zone of inhibition and record it in millimeters. Okay, so that's important. We're going to record it in millimeters. And the diameter of the zone of inhibition, let's say that this is one of our plates, and this is the iodine disc. Okay. So you recall that you swabbed your plates to get nice confluent growth. So when you pull your plate, you want to hold it up to the light, and what you're looking for is an area surrounding the disc in which you have no colonies growing. What is that zone called? Zone of inhibition, right? And you'll recall the theory behind this is when you place the discs on the agar, the discs that have been soaked in the different chemicals, the chemical starts to diffuse away from the disc, setting up a concentration gradient, right? So if the chemical can inhibit the growth of the microbes won't grow, but at one point probably the concentration of the chemical is so low, now the microbes are no longer inhibited, so they start growing again. What we're going to do, and there's many flaws to this technique, you guys, but just to kind of get a feel for this, what we're going to have you all do is we're going to have you measure the diameter of the zone of inhibition and report it in millimeters. Okay, so I'll just make this up, you guys. Let's say this is 25 millimeters. We want you to record the diameter of the zone of inhibition in your lab notebooks, and also we're going to put up a lab data sheet on the table. If you would have one volunteer from each team come up and record the diameters of the zones of inhibition, it will generate a class discussion that will be important for the quiz. Okay, so if each team could use just a different color chalk, that way that's how we'll keep the team separate. And again, you guys, I know everybody's tired, but the sooner we can get this done, the sooner we can move on to the last experiment and we can get out of here a little bit more efficiently. Yesterday, people were so exhausted. You could just, and I've been there myself, you're just sitting there going, can't move, can't think, you know. But if we could, as a team, tackle this and get these measurements done, then we'll be able to move through the rest of the experiment today. Okay? All right. I'll put the table up while you guys are working.